Hello everyone, my name is Carolina and today we'll be talking a little bit about mitosis. Cell division is a very important part of our life since it's responsible for transforming us from a little egg to a baby, adults, and also for repairing tissues and renewing and making life possible. The length of time that each cell will take to complete this process will vary. But before starting mitosis, the cell might be ready to complete all the steps that we're talking about today. The cell cycle involves the interphase, it corresponds to 90% of the time, and the process of mitosis and cytokinesis. Cytokinesis is not considered a part of mitosis, however, we don't know yet exactly when this process starts. During interphase, the first step is G1, where the cell will grow. Then we'll have the S phase, and we have the cell replicating the DNA, making a copy and making everything ready for the mitosis process. During the G2 phase, the cell is still growing and we'll have the final preparation and getting ready for division. During the interphase G2, we can see also the centrosome with two centrioles, because the idea is after mitosis complete, we will have two new cells, two daughter cells. So we might have the separation of the centrosome during the process of mitosis. The cell might show the chromatin, the DNA replicated, ready for division. We still can see the nucleolus and the nuclear envelope completely embracing all the chromosomes, chromatins, and nucleols. So what is mitosis? Mitosis is a type of cell division in which one cell, the mother, divides to produce two new cells, the daughters, that are genetically identical to itself. The goal of mitosis is to make sure that each daughter cell gets a perfect, full set of chromosomes. What are the phases of mitosis? Mitosis consists of 5 basic phases: the prophase, prometaphase, we see here the metaphase, anaphase, and telophase. Cytokinesis is not a part of mitosis process, but very important because it's where we are going to see the cytoplasm division. Before starting the mitosis process, the cell might get ready for division. During interphase G2, the cell has two centrosomes, each with two centrioles, and the DNA has been copied, duplicated, but they are not condensed yet. At this stage, the DNA is surrounded by intact nuclear membrane or nuclear envelope, and the nucleus is present. Prophase. The prophase is the first step of mitosis, and we'll see the mitotic spindles here separating the two esters that is composed by the centrosome and the centrioles, two pairs of centrioles. The mitotic spindle begins to form and it's a structure made of microtubules, a strong fiber that are part of the cell skeleton. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The centrosomes move away from each other, where the chromosomes start to condense, making them easier to pull apart later. So we can see during prophase that we have the DNA condensed. It's the first time that you can see the chromosomes, and they are attached to each other at one part that we call centromere. The two sisters chromatid find each other and they will attach themselves to the centromere. The nucleolus starts to disappear so we cannot see the nucleus anymore, and the nuclear envelope starts to break down. Prometaphase. So we can see here, prometaphase. During prometaphase, the nuclear envelope is completely gone, and the chromosomes are fully condensed, and we can see the connect core. The connect core are the microtubes that will attach from for the centromere, the part where the two sister chromatids attach to each other. And we can see also the non-connect core, that it's it will be responsible for elongating the cell to make it grow, and will not attach to any centromere. And then we are going to metaphase. Metaphase, the third step of the mitosis. We will see the spindle, the microtubes that are completely attached to the centromere, but capture all the chromosomes and line them up at the middle of the cell, getting ready to divide. So we can see here that all the chromosomes are, all the chromosomes are lined up in a place that we call metaphase plate. Basically, metaphase, it's the lining up of the chromosomes and getting ready to divide in anaphase. We can see also the centrosomes in each pole of the cell, and anaphase. In anaphase, the sister chromatids separate from each other and are pulled towards opposite ends of the cell. The microtubes that are not attached to chromosomes push the two poles of the spindle apart, while the connect core microtubules pull the chromosomes towards the poles. The chromosomes separate, and we'll see that they will become a daughter chromosomes. So, in anaphase, the sister chromatids separate from each other, and they are pulled from opposite sides of the cell. And the idea is to separate equally these chromosomes to get ready for division to the last phase of mitosis, that is telophase. Telophase is the opposite of prophase. Everything that we had in prophase, here we are trying to do the opposite. The spindles will disappear. The mitotic spindle is broken down into its building blocks. A nuclear envelope starts to reform around each set of chromosomes. We will have the formation of the nucleus form, and the nuclear membranes or the nuclear envelope will start to reappear. The chromosomes also starts to be less condensed. It was really condensed in the beginning prophase to get ready for division, and then here the chromosomes starts to be less condensed. In telophase, the cell is nearly close to divide, and it starts to establish its normal structures as cytokinesis takes place. Cytokinesis is not a part of mitosis. However, the division of the cytoplasm will finish the process of having two new cells. It may start in either anaphase or telophase, but the truth is we don't know yet. So the cytokinesis will act around the middle of the cell, pinches inward in cell animals, and we will see the cleavage furrow. However, In plant animals, the cell plate forms down the middle of the cell, so we will not see the cleavage furrow, but actually you'll see the cell plate. When division is complete, it produces 2 daughter cells. Each daughter cell has a complete set of chromosomes identical to that of its sisters, and the daughter cells enter the cell cycle again in G1. So in the beginning, when we were talking about the cell cycle, the interphase is the process that will take longer than mitosis. When division is complete, we will have 2 daughter cells. Each daughter cell will have a complete set of chromosomes. When cytokinesis finishes, we end up with 2 new cells, and each with a complete set of chromosomes. The daughter cells can now begin their own cellular lives, and depending on what they decided to be when they grow up, they may undergo mitosis themselves, repeating the cycle, getting inside the G1 of interphase. Thank you everyone. I hope you enjoyed this video. Bye.